Hi everyone, in this lesson I'm going to teach you about streak plate technique. Streak plate technique is a very important microbiology technique. So we use it all the time as microbiologists when we work with bacteria. Here in this picture that I'm pointing to, this is an agar plate. That's what we grow bacteria on, and there's bacteria growing on it. With this method, which I'm going to explain in a little bit, we streak bacteria so that ultimately we get these little circles that we call colonies of bacteria. So streak plate technique is a technique that allows you to streak bacteria on a plate, on a petri dish, an agar plate, so that you can isolate colonies of bacteria. These little circles are what we call colonies of bacteria. A colony of bacteria, that little circle, is millions of bacteria that started from one little bacteria. So you cannot see a bacterium one with your eye. It's divided so many times that every circle is millions of bacteria, and it's a pure culture. So the goal is to dilute your culture enough to get these isolated colonies, and isolated colonies are pure culture. So every little circle is a pure culture of that bacteria. And this is a very important technique, especially when we're working with mixed culture. A mixed culture of bacteria means that you have multiple species of bacteria, not just one. So a pure culture, for example, would be if I have like a test tube of just E. coli. A mixed culture is E. coli and Salmonella and Proteus and Klebsiella and so many other bacterial species. So anytime we have one bacterial species, we call that a pure culture. Any more, that's a mixed culture. If you have a mixed culture of bacteria, so imagine I had a test tube and I want to isolate what's in it, I would do streaked plate technique to isolate individual colonies. And if you look at this plate is a mixed culture, but we see individual colonies. So there's a yellow colony here, there's a red colony, there's an orange colony. If I wanted to do more research on just this yellow colony, I would get the sample from here and do more experiments. But you can only see colonies if you do streak plate technique so that you can isolate pure individual colonies. And the way this technique is done, I have a video showing me in lab doing it, but basically what you do is you get a sample of your culture. Your culture is in a liquid broth, and you streak it on an agar plate, and then you do not go into your original culture anymore. You sterilize your loop, and then you take a little bit from that quadrant and you go into a second quadrant, streaking it. So I'm diluting it as I go. Then I sterilize my loop again on the flame, and then I go into this culture one more time. Sorry, this streak area one more time, and I streak in the third area. So I do 3 quadrants. So the first area that I streaked has the most bacteria, then the second area is getting diluted a little bit more, and then the final area we see colonies on. So it's progressive streaking so that we can dilute our culture enough to get these individual colonies. And again, a colony is millions of bacteria that evolved from one species. So the things I want you to know from this lab is I want you to know the steps in streak plate technique. So how we're doing 3 quadrants and how you go from the original culture and you do your first streak, then you sterilize your loop, and now you're only using your plate. Then you do the second quadrant and the third quadrant. The goal of streak plate technique, so read your lab, the goal of it, remember, is to isolate single individual colonies. A pure culture is just one species of bacteria, and a colony is millions of bacteria that is originated from one species of bacteria.